Hello, and welcome to this webinar presented by the IET's Cambridge Network. We represent members in the Cambridgeshire area, but we're very pleased to welcome members in other parts of the UK and indeed around the world. My name is Phil Zernghast. I will be the chair of the event tonight, and I'm also the chair of the local network. Billy, can I just ask you to switch off your screen sharing for a moment? Before we get underway, I'll just mention some housekeeping points. We will be taking questions, but if you'd like to post some, please use the Q&A button, not the chat button, please. We can't answer anything on the chat panel. Our speaker will try and answer all the questions at the end of the presentation. If we don't get through them all, we will keep a record of those, and he's offered to answer those offline later. We'll be recording the event tonight, and that'll be available later on the IET Cambridge YouTube channel, which you can find by searching for IET Cambridge. The IET has asked me to mention that this year it's running a diversity survey to better understand the characteristics of its audience and how best to serve them. So I will post a link to that in the chat panel later and at the end of the presentation as well. Finally, I'll just mention our next event, which is coming up on the 15th of April, which will be on the subject of electric aircraft by a company called Vertical Aerospace. Now, on to tonight's speaker, and I'm very pleased to welcome Billy Boyle, who will talk about Owlstone Medical's Breath Biopsy platform. Billy has a master's degree in engineering from Cambridge University and is a co-founder of Owlstone Incorporated. Now, Billy helped develop the core chip technology, which was commercialized also by Billy for use in the scientific and industrial markets. But more recently, he's focused on medical applications and working with clinical partners. They've explored the application in early stage cancer diagnostics. Now, in 2016, Billy spun out Owlstone Medical from the company and is the founding CEO. And in 2020, he was made a fellow of the Royal Academy of Engineering. So without further ado, I'm very pleased to hand over to Billy Boyle.
Brilliant. Thank you for that, Phil. Can I just double check and see the screen all right?
I'm seeing the screen and I'm hearing you very well. So
thank you. Thanks a lot for having me tonight. So really delighted to be here. So our company, Owlstone Medical, is developing a technology called Breath Biopsy. And we set up the business to try and save 100,000 lives. So we see two big problems in healthcare. The first is that too many people are diagnosed with disease when it's too late to cure. And the second is too many people are on treatments that don't work for them. So there's probably no better example of this than in cancer, where we know if you can pick up cancer sooner, survival chances are about 10 times better, but also treatment costs are about 10 times lower. So that's really what we're about as a company. And as Phil mentioned, I'm an engineer by background. So I studied at Cambridge, stayed on as a researcher there. And my two friends at Cambridge, David and Andrew, we both worked in the same microsystems and MEMS group. And we got very interested in chemical detection. And that was the kind of original focus of the technology that we spun out into Owlstone. And what I was hoping to try and do in the talk today is, go through the technology in a bit more detail, what it is that we're trying to do as a business, but also provide a little bit of a kind of unvarnished history of some of the challenges along the way in the company. So the core technology is a programmable chemical sensor. And what makes it special is you change what you detect just by changing the software on that. And when we first spun out, our original focus was around security and industrial applications. And over the years, we raised quite a lot of VC financing. We won quite a lot of government grants, and that allowed us to both mature the technology, but to build it into a range of different products. And as Phil mentioned, around 2016, we got really interested in the diagnostic applications, and that's when we spun Owlstone Medical out as a completely separate business. And today we've got a team of about 170 based over at the Science Park in Cambridge, and we've raised close to $100 million of investment. And I think there's three key things that we're focused on. So I'll describe the underpinning technology in a bit more detail. But when you breathe out, you have thousands of chemicals on breath. And the heart of our breath biopsy technology is the capture and analysis of those chemicals, and using our sensor technology and lab-based instruments for those types of analyses. And on the one hand, what we're trying to do with the technology is develop and launch tests ourselves, so particularly around things like early detection of lung cancer, colorectal cancer, precision medicine, respiratory disease. But actually, we see breath as a completely new diagnostic category. And we can't explore all potential applications of it. So what we've also done is made these products available to the very broad research community. So academic and pharma and clinical researchers who want to try and investigate whether or not there's breath biomarkers for their particular application, they can use the technology to do that research themselves, and we collaborate and partner with those types of organizations. And then the last bit is looking at the data that we can get. So both through the trials that we run and the trials that we run for our partners, we see a huge amount of opportunity and excitement in being able to collect these data sets to help try and accelerate the pace at which you can discover novel biomarkers across a broad range of different diseases. So I think, the initial slides, showing the team as it is now, and, talking about how we raise capital kind of makes it seem like it was all kind of easy and straightforward, and, nothing is further from the truth. So when we first got interested in chemical sensing, so we were, as I said, engineers by background, but we had no clue about the commercial aspects of, how to start a business, how to sell things, how to raise investment. And we were quite fortunate in and around the Cambridge area. There was quite a vibrant student entrepreneurship scene. So there's a lot of support and mentors, who were very willing to help. People had an idea but didn't really know anything about the commercial aspects of starting a business. And I remember I think it was the first or the second pitch that we ever did to an investor. We were presenting. The investor said, what's your margin? We kind of look at each other and we're like, we don't know what margin is. So we had a bit of a guess and hoped that more was better. So we're like, our margin is good, our margin is good. And then off we go and, search up on Wikipedia or Google what margin actually means later. But again, I think we were so lucky in those early days to, have the support of people who have been there, done it before, to help get us, spun out and started. So back in 2004, actually on the day this picture was taken, this was the day where we raised our first $2 million worth of investments, and we were off to the races. So we knew what we needed to do technically, but I don't think we knew quite how hard it was going to be. So the heart of the sensor technology, and because this is an IET talk, I did want to talk about it in a bit more detail. It is this reprogrammable chemical sensor, and it acts a bit like a filter. So if you think about the challenge in any detection problem, be it detecting explosives, toxic gases, contamination in wastewater, or looking for biomarkers in breath for disease, you have a mix of different chemicals together. And what you want to try and do is filter out those chemicals which are not of interest and target those specific chemicals that you're interested in for your application. So that's fundamentally what we're trying to do with our chip. And the chip is a MEMS-based device or microsystems-based device. So as well as electronic functionality, we have a mechanical structure in there. And typically what we do is that we will have a mix of different chemicals coming in. Those chemicals are ionized, which means they can see an electric field. We flow them through our MEMS chip here. And when you look at the cross section through the MEMS chip, it's a deep etched silicon device. So etched the whole way through the silicon wafer. And as the ions move through the device, we apply electric fields in such a way that we can tune it to allow the particular ions or chemicals of interest to pass through and then be detected. So if you were to zoom in on one of these channels here, as an ion is moving through the filter region, we apply electric fields in such a way that we filter out those things that we're not interested in, and our target compound passes through the filter region. So we're applying a waveform, and the ion is moving through this filter region, and that target compound of interest pops out the end and gets detected. So you can see this where we're applying these electric fields, this oscillating electric fields. For this chemical we're not interested in, it gets cancelled at the sidewall of the device. So within the silicon MEMS device, we have many of these channels etched the whole way through the silicon wafer. And then when you look at a flux of ions coming through the device, this is just a simulation that shows how ions move through the device as you alternate this waveform. What you get at the back are these target compounds. And again, this could be explosives, biomarkers, and breath, you name it. And what makes it so powerful is you never have to change the hardware. It's always the same hardware. All you're doing is changing this tuning parameter, which you're programming in the software of the device itself. What you end up at the end of this is a chemical fingerprint, if you like. That's how we think about it. So by changing these different parameters, you end up with a very information-rich fingerprint where the location of peaks and the shape of peaks are very characteristic of what a particular chemical is. So you can see here different types of examples from some of the industrial work that we've previously done in the past, where you have these very distinctive fingerprints for the different compounds. And what the team have been doing recently, which is very exciting, is looking at these information-rich chemical fingerprints and applying machine learning, artificial intelligence algorithms to effectively learn what these different chemicals look like, even in quite complex backgrounds. So you can quite quickly build robust classifiers for these different types of applications. So that's just a little bit about the core sensor technology. I'll move on to talk a little bit about how we use it in different types of applications. I think, again, one of the things that we saw quite early on was that we were positioning ourselves initially as a sensor company. We'll make the sensor, other people will integrate it into the product. But the problem that we found was actually, people wanted an end system. So we had to move up the value chain and start to make instruments and products ourselves. And we initially were very focused on security and defense applications. And that was partly because after 9/11, there was a lot of funding going into better sensor solutions for toxic gases and explosives. And also, it was a very good way to get non-dilutive funding to mature the technology where you had a ready customer at the end of that process. And we matured the sensor technology. We built it into hardened products like you see on the right-hand side. And these systems have been deployed in various locations in the world. But one of the things that we were trying to do was, map out what are the different applications of the technology. So there's the security, industrial, ultimately diagnostic type applications. We knew that was the furthest one away, so we had to figure out a way how to get there. And in 2007, 2008, part of the difficulty in this business was, during the Great Recession, all the funding evaporated. So, venture capital funding evaporated, but also a lot of the defense and security contracts as well evaporated. So we had to pivot quite quickly and think, okay, well, we've got this core technology working. What are the other applications we can use it in? And so we started to sell into industrial process control. So this is looking at things like contamination in crude oil, so things like methanol in crude oil, contamination in wastewaters, And this was great because it gave us, a market that we could sell into, where we could keep the business sustained and going. And one of the things that was quite interesting around that time was because the sensor technology is general purpose, you can look for different compounds. The academic research community became more and more interested in using the technology in their research. So this was around things like medical diagnostics, where there had been a lot of work or papers published reporting that there was chemical markers in breath and then urine for different types of diseases and illness. So more and more papers were being published using our technology. So we started to look at this a lot more seriously, thinking, well, actually, this could be a really significant opportunity and use case for the technology. And this paper was one of the transformational ones for us. This was one of our partners up in Warwick, who used the FAMES technology, our sensor technology, and our Lone Star products to look at the chemical composition in the headspace of a urine sample for patients who had colorectal cancer and those that didn't have colorectal cancer. And they got some pretty encouraging results from this initial paper. So we started to dig into this a lot more and say, what more can we do here? And around the same time, we were starting to think about different clinical problems and clinical needs. And this is, to this day, I think one of the most shocking statistics I've ever seen, that 1 in 2 of us are going to get cancer in our lifetime. From parents, partners, children, grandchildren, 1 in 2 will get cancer. And when you look across different types of cancer, the survival picture is very variable. So in some cancers, treatments are actually pretty good, or there's pretty effective screening tests already in place. But you look at some other cancers, things like colorectal and lung cancer, where they affect a huge number of people, and actually there's not been a lot of progress made. In lung cancer, there's been barely any progress made in survival over the last 40 years. And I think one of the things that to me was also quite shocking was that actually the solution is in sight, in the sense that if you can pick it up sooner, your survival chances are so much better. So for colorectal cancer, survival chances are about 90%, 95% if you pick it up at stage 1. And this drops to about 5% if you pick it up at stage 4. And so while this was happening, the work that we're doing with the technology and our academic partners, my own wife was diagnosed with stage 4 colon cancer. And that was, again, like, I was angry because if she had been picked up sooner, her chance of survival would have been so much higher. But because it was stage 4, she didn't make it. After 2 years worth of treatment, different surgeries, she died on Christmas Day in 2014. And, while for us as a family, we were heartbroken, the thing that was also clear was that, we were in no way unique. This is such a common story. The amount of people who are diagnosed late, and when you look at something like colon cancer, kills 215,000 people every year, costs $99 billion of investments, and actually, for colon cancer, it's probably better than other cancers because there's actually a screening test available for it. But part of the challenge is that even with the screening tests that exist today, too many people are still diagnosed at that late stage. And I think a lot of this comes down to the nature of the test. So, in a lot of cancers, there's just no test. And in something like colorectal cancer, where there is a test, you need the test both to be good, have high sensitivity and specificity, but you also need to have people show up for the test and want to do the test. And with fecal-based testing or colonoscopy, Often that's the problem. People just don't want to show up to do the test, and it's usually about 50% compliance rate on average. So what that means is 100 people have colon cancer, half of them just don't show up for the test in the first instance. And the second problem is the test isn't actually that great. So again, it's better than nothing, but it's not that sensitive. So out of the hundreds patients, you're not picking up actually that many cancers. So I think there's a lot of room for improvement. And I think this was obviously a big driver for us to say, what can we do here, and what role do volatile chemicals play? And I'm sure a lot of us on the call this evening will have seen stories of cancer sniffing dogs. It's in the news about once every 2 months, it seems, or COVID sniffing dogs. And actually, when you look at some of the papers that have been published, there's fairly reasonable evidence there. So, blinded validation studies where these dogs are able to pick up different cases. So, the $1 million question is, what is it? Why are they doing this? What's going on here? And actually, the origins of breath-based research stretch right back to Greek times. And when you breathe out, objectively, there's thousands of chemicals on your breath. And the question then becomes, what are these things, and where do they come from? So some of them are produced in the body. So if you think about metabolites within the body, some of these are volatile and they come out on breath. And this is interesting and exciting when you start to think about diseases such as cancer, which we know have an altered metabolism even at a pretty early stage. So you're trying to say, well, can we see some of these variations in these endogenous metabolites which come out on breath? And the second class of compounds that you find are exogenous. So things that originate outside the body, come in and then go out again. And normally you think of these as chemical noise, stuff you want to ignore, but actually we've pioneered an approach called evoke probes, where we deliberately administer compounds to the body to target specific diseases. And I'll talk a wee bit more about that. So I think breath has some, obvious advantages. The non-invasive nature of it. It's the ultimate non-invasive test, anytime, any place. So when you think about how to overcome some of the challenges of acceptability of testing the patients, it doesn't get better than breath. But actually, I think there's another really important and subtle advantage of breath. And that is that with each exhalation, you have these thousands of chemicals on the breath. And actually, what we do with our technology is with each exhalation, we trap all of those chemicals and you can enrich and pre-concentrate it over time. So just through normal tidal breathing, you're building up this the trapped concentration of the volatiles coming out from breath onto our collection device. So what this means is that about once every minute, your entire circulating blood volume goes around your body. So this is a bit like a window into that entire circulating volatile metabolism. So we think this is really an important advantage because it will help drive sensitivity in a lot of these early detection problems. That's fundamentally what you need if you're going to pick up disease sooner. So this all sounds brilliant. Because we were coming at this from a standpoint of, we had a business in security and industrial applications. So we're trying to figure out, is there something in breath or not? Despite all the things that we had seen, the raging skeptic in me was like, well, if it's so good, where are all the tests today? And the more and more we dug into it, the more kind of, basic, quite foundational problems we identified. So simple stuff like, how do you get a good breath sample? So that hadn't really been addressed before. And the problem is, if you don't get a good sample, you're not going to get the right answer, irrespective of how you analyze. One of the other things that we saw was that quite often you would have quite small scale clinical trials or pilot studies where chemicals of cancer or other diseases were identified, but they weren't validated in big independent test sets. And you need to do that to prove whether or not there's actually utility in some of these compounds which are being identified. And also, when you think about the basic discovery, often people would use the wrong types of analytical approaches in discovery, confusing it with what you need when you come to actual deployment of tests. So I think what we've been able to do as a business is address each of these big key limitations. I'll talk a little bit more detail about each of them. So the first is a receiver breath collection device. And this effectively allows you to capture and trap all the volatiles from your breath into a cartridge. And once it's on this cartridge, it's completely stable. So you can send it back to a lab for analysis. And I've talked already about our programmable sensor technology. It's super fast, very sensitive and selective. Can be deployed point of care. But actually, one of the problems that we found was that for this technology, you need to program it for what you're looking for. So in the security application, it's pretty straightforward. You say, I'm interested in these explosives, and you program the chip for those particular explosives. But actually, the million-dollar question in a lot of these diagnostic tests is, well, what are the VOC biomarkers for something like lung cancer, for instance? And I'll talk a little bit about how we try to address some of those challenges. And then this last point here is the use of, rather than just relying on what comes out of the body as a way to diagnose disease and illness, can we introduce specific probe compounds that are going to target different diseases and actually give us a much larger signal compared to what you would normally just get out of the body? So the receiver device, here's just the product in a bit more detail. It allows you to do different things. So, one of the things that became pretty clear to us, at the outset of the COVID pandemic, was that the world now knows about the importance of breath as a sample matrix, both for what's contained in the aerosol and the respiratory droplets, so things like viruses, bacteria, and proteins. But we also have these volatiles that come out as well. So we're interested in all of those things. So we're interested in the volatiles, but we've also been doing a lot of active research on that aerosol respiratory droplet side of things as well, because it's very pertinent to COVID, but it's also pertinent to a lot of other types of infectious diseases and illnesses as well. So again, with this device, we've solved the sample collection problem, and that's a biggie because that's the kind of entry ticket to the party. Unless you do that, you don't get higher quality data down the line when you're doing your discovery type analysis. And I kind of alluded already to one of the problems that we started to see and observe when we first started to get involved in the field. I think in some ways we actually thought, oh, this is going to be quite straightforward. A lot of papers have been published reporting different chemicals and things like lung cancer. We'll just take those chemicals and program it into our chip and we'll have a test. But again, what we were finding was that all these studies were really small and actually none of them were being replicated. So you compare like two different lung cancer studies and they're saying different things for what the most important chemicals are. So we realized you needed to bring it back to basics and say, actually, the challenge is around the core discovery and validation of some of these VOC biomarkers. So when we do analysis, we think about it in two ways. So, we have a technology that once you know what you're looking for, you can program. But actually, if you want to be doing, robust discovery type workflows, you need to invest very significantly and heavily in, top of the range GC mass spec capability. So we've done that over the years. We've built a breath biopsy lab, which is kitted out with some of this high-end instrumentation. We have a partnership with Thermo Fisher there. And this is all about trying to do that initial identification of what the most important biomarkers are. And once you know that, you're off to the races. You can program it into our technology, other mass spec instruments as well. And that's where you're quite close to having a test. I just want to introduce the concept of evoked probes a little bit more, because I think this is really where a lot of our focus and emphasis is in terms of how we are thinking about breath diagnostics, both in cancer and also things like liver disease. And the basic principle here is that when you introduce a compound into the body, you can target different organs or pathways in the body. So for instance, you can introduce compounds that will be metabolized in the liver, and that means you can start to think about applications looking at liver disease, liver function. And what we also think quite a lot about is in cancer, particularly in early cancers, what's different about a tumor compared to healthy tissue that allows you to introduce an evoke probe that is then going to be detectable on breath. And the kind of analogy or the mental model that we have for this is around cancer imaging. So if you think about established techniques like a PET scan, where you introduce a substrate into the body, because tumors have an altered metabolism, they have an enhanced uptake of that substrate. And that's what you see when you image. You see the enrichment, the enhanced uptake of that substrate within a tumor. And this demonstrates two things. One, that a tumor does have altered metabolism. So if you're thinking about differences in composition of chemicals in breath, there's a feasible mechanism there. But it also highlights the principle of introducing a probe compound into the body. But for us, we look at this and say, well, can we use probe compounds, but rather than put someone into a $20 million scanner, can we simply release chemicals out onto the breath if there's a tumor present or not? So we're running a number of clinical trials at the moment. And again, we were very, very fortunate. CR UK have helped us a lot, connecting us to the right investigators, looking at different tumor types. They've been able to help us, get patient samples and run clinical trials. And I would say a lot of our focus at the moment is around lung and liver. And looking at the evoke probes in things like liver disease and liver cancer, the specific compound that we've been looking at over the last couple of years is a compound called limonene. So limonene is a very safe compound. It's commonly found in foodstuffs. You'll find it in citrus peel, for instance. And in the past, we used to think of things like this again as chemical noise, stuff you just want to ignore if you see it on the breath, because it's not being produced in the body. But actually, limonene is quite interesting because once it gets ingested into the body, it's metabolized in the liver by these CYP enzymes. And actually, if you have a diseased liver, you metabolize that limonene differently, more slowly. And limonene is lipophilic. That means it goes into the fat compartment in your body. So the hypothesis here is that over time, as you're being exposed to limonene, if you have liver disease and liver cancer, you should have much higher levels of limonene on your breath compared to someone that's perfectly healthy, who's able to metabolize that limonene without any problems. And we ran a clinical study with Cambridge here at Addenbrooke's, and that data was published just last year. And that's indeed exactly what you see. For people who are healthy, you have very low levels of limonene, and then patients that have liver disease and liver disease complicated by liver cancer have much higher levels of limonene. And this seems to scale with the severity of the disease as well. So this is a really exciting finding, and that's allowing us to push forward and think about the next phases of these clinical trials, where we can introduce very specific quantities of these evoke probe compounds such as limonene, but we can also combine them together. So rather than just rely on a single probe, you can put in multiple probes at the same time, target different enzymes, and get this readout on breath, all completely non-invasively, pain-free. So this is really, really exciting result for us. And then I just wanted to talk about the second part of what we do as a business, because it also feeds very closely into our company mission of how we have an impact on patients. And this basically comes down to the fact that, you know, we have a technology platform we can use in lots of different ways, but we fundamentally are resource limited in terms of the amount of people that we have, the amount of money that we have to invest. So we try to focus on things like liver disease, like lung cancer, where we know for sure there's a big unmet clinical need. But at the same time, we want to, you know, democratize this technology. We want to get it out there into the hands of as many people as possible, so they can do active research and say, you know, what can we do with breath? What are the other biomarkers and other clinical applications that we can solve? And typically what this means for one of the studies, or, you know, one of our academic partners, is that they get to receive a device and they collect breath samples from the patient, and this could be patients that have disease, some controls as well. And rather than them having to invest in, you know, all the infrastructure and the, you know, hugely expensive instruments, you send the sample back to our lab and we'll do it for you. So that is a very cost-effective way for them to access, you know, very expensive high-end instruments. And what we also have is, you know, a fantastic team internally that can help with the data analysis, you know, understand what chemicals are coming out and how these relate to different biological pathways, for instance. So it's really allowing these researchers to get the most out of these studies in the lowest cost way for them. And we've had a lot of really exciting success over the last couple of years or so. So working with a lot of pharma partners, you know, contracts like GSK, AstraZeneca, J&J, but also big groups like Cleveland Clinic and the Mayo Clinic. But I think what is most exciting is that, you know, having completed these studies, or these partners who have been using our technology, our samplers or analyzers, they've started to publish more and more papers that show the, you know, the real potential breadth of applications for breath biopsy. So this isn't just cancer. This is, you know, inflammatory diseases, cardiovascular disease, infectious disease. So all these papers and publications, you know, they're all up on our website. And then what we try to do is to say, okay, well, where, you know, of these different papers, where is there real clinical needs, and where that data looks really good and really promising, where we can then put our own investment behind it. And there's some, you know, really wonderful examples of this. You know, one of our you know, fantastic academic partners down at Imperial has published papers looking at volatile biomarkers and breath patients that have pancreatic cancer. They've also done exciting work using the technology in esophageal cancer as well. And what was really nice about some of this work was that they were looking not just at the chemicals on the breath, but they were also looking at, you know, when you took a tumor out of someone after surgery, looking at the chemicals coming off the tumor itself, and then trying to match these two and say, well, if the tumor is producing these other chemicals, is that what you actually see on breath? And again, what's great about a lot of these papers and publications is we can look at these compounds and then we can say, okay, here's a hypothesis. You know, let's go and check to see if we can find these compounds in much, much larger studies. And it's in those much, much larger studies you fundamentally need to run if you're going to get tests accepted and onto the marketplace and being used day in day out by patients. And then the last example here, this was another customer of ours in Italy who really recently, I think it was only 2 months ago, 3 months ago, published a paper using our receive device looking at colorectal cancer patients, showing again very encouraging early stage data our view is always, you don't believe stuff until it's been independently validated. But again, it's really, really encouraging at this stage. So we're very optimistic that when you look at the problem of early cancer detection, my view is someone just needs to solve it. This needs to be a solved problem. And I think one of the most exciting things that's been happening over the last 4 or 5 years is the amount of investment and the amount of different technologies which are being developed trying to tackle that early detection problem. And Cambridge particularly has a huge focus on early detection. I think of everything that you could do in the cancer space. If there's a single lever that you could pull that would be most transformative for maximum amount of people, it'd be picking up the disease much sooner. So I just wanted to finish off with the last couple of slides about thinking about what else can we do. And I think a lot of our emphasis is around the cancer detection, but we do fundamentally see this as a much broader range of applications. And the heart of that in some ways is getting high quality data sets. So by running trials, ourselves or with our partners, by knowing whether or not those patients have a particular disease or a healthy control, other comorbidities, we can start to build quite unique data assets. So well phenotype patients and all the compounds that we can analyze on their breath. And this is very exciting because I think it will dramatically help in the discovery of novel biomarkers across a range of different diseases. And ultimately, our hope is that in the not too distant future, breath will be a tool that sits alongside and complements what you can do with blood or urine-based analysis. So it's not going to completely replace those things, but it will be able to do things that you can't do with those types of samples. And crucially, I think, again, because of the patient acceptability, you'll have a much higher rate of uptake for different tests because people won't be afraid. They'll be happy to give the test because it's completely pain-free. So our hope is that Owlstone will be the leading company within the breath space. So in a few years, if you go to a GP surgery and you're collecting a breath sample, our hope is that it's going to be our technology that will be in that GP surgery. But for me, everything comes back to why we started this. What is it that motivates us day in and day out? We believe we can create a new industry, but actually it's the application of science and technology to try and make tests that will save patients' lives. So that's what motivates me. That's what motivates our team. And I'm very fortunate that we have a real world-class team that are solving really hard problems day in and day out, and we won't rest until we meet our company mission of saving 100,000 lives. So thanks again for the time and the opportunity to present. I've left enough time, hopefully, for us to have the chance to answer a few questions.
Thank you very much, Billy, for an excellent presentation. So I will now hand over to David Blake, who will take us through the Q&A session. David.
Well, thank you very much, Billy. It was certainly an interesting session, and you've got quite a few interesting questions. There's quite a range of questions, ranging from some about the technology and also to some about the capability of what the systems can do. Perhaps we could start, though, from the beginning. Somebody's asked, how does food consumption affect the detection of markers? In other words, if you get something in your stomach, is that going to affect what you can measure?
Yeah, no, that's a good question. So on one of the earlier slides, where when you look at the chemicals in the breath, you have a mix of endogenous and exogenous. So endogenous is the compounds being produced in the body. So things that come in through dietary intake, but pharmaceutical drugs, things you've been exposed to, environmental pollution, for instance, they will come into the body, and some of those will be coming out on the breath as well. So actually, in a lot of applications, that's just stuff you want to ignore. It's not relevant to the diagnosis. So you just need to make sure that you have the analytical specificity to be able to resolve the peak of interest from whatever, if you had lots of garlic in your dinner, for instance. But actually in some applications, that's what you're really interested in. So if you think about things like environmental exposure, for instance, and disease more generally, in disease, genetics loads the gun, but environment pulls the trigger. So actually, we're seeing more and more applications where people are interested in these chemicals that we're exposed to every day through things that we breathe in and things that we ingest, and what they're actually doing to us.
Oh, great. There's a question here of a particular specificity. Can this system detect early signs of stomach cancer?
We are not doing any research on that ourselves. I'm trying to think if any of our academic partners are. I don't think so. I think we've done mainly upper GI and with some of the work I show on the colorectal cancer as well. Like the one thing I'd say is that the mechanisms of how endogenous VOCs are altered is generally true across different solid tumor types. And actually, when we use our Evoke probes in early cancer detection, some of the mechanisms that we look to try and target with the Evoke probes also tend to be common across different solid tumors. So, we're very focused on lung cancer at the moment, but if we have success there, then it's reasonably likely that you could apply it to a much broader range of solid tumors.
Okay. There's a question actually about the sensor. Do you use ionization in your device? And if so, what mechanism do you use for gas ionization?
Yeah, we do. So when you introduce your gas, you need to ionize it in order for the ions to see the electric field essentially and be filtered. So over the years, we've used 3 different types of ionization, and it depends a little bit on the application that you're interested in. So we use things like nickel 63, which is a beta emitter, so ionization or a rad source ionization. We've used UV ionization as well, and corona discharge. So we've seen, for instance, in some of the industrial applications, if you're interested in something like benzene, you tend to want to use something like a UV ionization source, because with a nickel ionization source, it's actually quite hard to ionize benzene if there's a complex background. So that's an important step. When you think about how the sensor works, there's kind of 3 steps. There's, generate your ions, then you filter, then you detect. Actually, the filter and detect is relatively straightforward, but if you never generate your ion in the first instance, you're not going to see that thing. So we do think quite a lot about the ionization source and the ionization chemistry for particular applications and compounds of interest.
Okay. You spoke, in fact, about the filter system inside the device. There's a question here: is it fair to assume that the effectiveness of detection of fingerprint is reliant on appropriate flow rate? That's obviously through the filter.
Yeah, so there's a bit of a balance. So if you push the ions through with the flow, and at a lower flow rate, you tend to have sharper peaks. So that helps the resolution and the selectivity. But you have smaller peaks as well. So that adversely impacts the sensitivity there. So again, you're always trying to find the sweet spot for your particular application. So flow is one of those parameters that you can play with to make sure that you're getting sufficient sensitivity. So if you need to detect at 1 part per billion, you can do that, but you're ensuring that you get maximum separation between the other stuff that might be present in the background.
Yeah. Are all chemical markers unique, or can one marker have several causes?
Yeah, no, that's a good question. So yes, there's definitely situations where some compounds can look quite similar. It tends to be worse in other more basic chemical sensors. So things like metal oxides sensors and enoses and stuff like that. You have a lot of cross-reactivity or things that look quite similar. I think one of the big advantages that we have is the vector that we generate has such a high information content within it. So you're looking at where a peak may not be fully resolved in one location, if you increase what we call the dispersion field, which is the intensity of the field that you apply, then you can resolve the peak from each other. So I think it's definitely something that can happen, but we have a lot more flexibility to deal with that, because again, you can change some of these applied fields parameters to try and get better separation.
Good. There's a question about a specific disease, and that's detection of Parkinson's. Apparently, there's been many recent reports that Parkinson's can be detected by smell. This is something that Owlstone may look at.
Yeah, no, it's fascinating. There was actually a paper published today, and it was in the news today. So it's a group in Manchester. So it's a researcher we know very well, Purdy Barron. She's done brilliant work there. So it's looking at it's a swab sample, so it's the sebum that you're looking at. And what was interesting about that, kind of similar to the dogs, if you like, was that it was a lady there who, she said, I can smell Parkinson's. And that's what triggered that research group to start to use mass spectrometry to try and figure out what those chemicals actually were. And again, I think that's a really good example where you go from that initial observation to really quite solid data that's saying that, yes, it looks like there's some volatiles or semi-volatile markers present that can help in diagnosis there. Like, I think part of the, on the one hand, you're trying to, robustly identify those markers, but you also need to think about how that's going to be translated to routine use. If you go down to a GP surgery or if you're in a pharmacy, how are you going to do that type of thing? So it's difficult to see how the dogs would translate to routine use. But what was really nice about that work was it's fairly simple to do, just a swab and put that in a vial and send that back to the lab for analysis. So that's, we're not working on it directly ourselves, but we're very familiar with that work, and I think that's one of those ones that's really quite solid.
Okay. On a more practical issue, to get away from the science for a bit, how is the device sanitized after use? Particularly with COVID, any contagious respiratory disease might sort of transfer. So how do you go about that?
Yeah, no, that's a very good question as well. So when COVID first hit last year, like, on the one hand, what we realized was, the world now does see the importance of breath as a sample matrix, but equally what we realized is people are going to be very, very concerned about this, and we need to be absolutely certain that you don't have any type of cross-contamination risk. I think one of the things that was quite fortunate was that even before COVID, that was something that we designed for anyway. So there's other respiratory viruses as well that you want to make sure that you're not contaminating the base unit with. So what we have on the receiver device is a reusable portion, and then a portion which is essentially thrown away. So the bit that actually touches your face that you breathe into is a silicone mask that gets thrown away, but it's got an N95 filter in it. So the type of filter that you find that you wear in a hospital if you're in a COVID ward, for instance. So actually, if you're breathing out, it's probably better for anyone else that they're breathing into this N95 filter, because it's capturing the respiratory droplets compared to them just normally breathing within the room. But we also did a lot of, we have a decontamination protocol, and that was all validated through external lab testing as well. So that, absolutely something that we make sure with our partners they follow those decontamination protocols.
The more practical question is, what is the approximate cost of this sort of technology? I don't know if they want to buy one off of you, but I think it's probably more to get a feel for the comparison between these sort of devices compared to sort of more complicated clinical systems.
Yeah. So at the moment, what we sell is the research products and the research services. So we don't have tests that you can routinely use yet, because you need to find those biomarkers first. But one of the things I think that's interesting about breath and the type of analysis we do compared to other things like liquid biopsy. So liquid biopsy is, lots of investment going into that for early cancer detection. It's actually still pretty expensive. So you're looking at, probably hundreds of dollars for a test there. So we think that, once you know the compounds of interest and you're running routine analysis, actually the cost of a breath type test can be in the low tens of dollars. So much lower price point compared to some of the alternatives, and compared to imaging as well. And we think that's important because if you look at, cancer is a global disease. So if you look at lung cancer in China, huge prevalence there. You need, I think, three basic things to be true. One, the test has to work. You have to demonstrate you've got sufficient sensitivity and specificity in the intended use. Two, people must show up to do the test. They have to be okay with doing the test. And that's why I think breath kind of knocks the socks off everything else, because it's easy. And then the last bit is around that cost structure, that price point. Like if we want to have the biggest impact with people, one of the things that we need to kind of push for is to make sure it can be as accessible by having as low of a price point as possible, but also allowing us to be successful as a business. So I think you can definitely do that with breath biopsy in a way that might not be as easy with some of the other approaches.
Okay. You've got a massive load of questions, which you're going to be here to midnight to answer them all. But so I'll try and move on to some others. How far away is one of these disease tests from becoming fully validated and deployed?
Yeah, so I think when you look at the different diseases, they have different risks associated with them. So the cancer problem is the hardest problem by quite a long way. So there's no guarantees that will be successful, but our kind of belief is that you have to try. The problem has to be solved, and if there's a marker in breath, we will be the guys to find it. So I have utter conviction about that. We've already launched a research use only panel in respiratory disease. I didn't really talk about that in the presentation earlier tonight, but that's looking at how do you better steer patients to the right therapy in respiratory diseases such as asthma and COPD. So that is out there now. We're targeting the pharma customers in the first instance for that, because again, it's a way to build up the evidence base. But also we will have our first test on market that will be used by patients, day in, day out in the UK this year in digestive health. So that's the plan. That's the first one. And then hopefully, you get the first one out the door, and over time we can add in more diseases as we find and validate more and more biomarkers.
Okay. And can this system then be used to detect virus particles? I think that's because of the COVID issue at the moment.
Yeah. So the you can capture, it's actually the same N95 material. You can capture the respiratory droplets of the aerosols that contain the virus, and then you can extract and analyze that. And actually, one of the bits of research that we were doing was to say, is this better than collecting a sample from a swab? Because the swab's not the most pleasant thing. So it is feasible, but we don't think the performance is as good as something like a swab. I think there's a more interesting question, which related to COVID as well, which is for the VOCs. So as well as the virus on the respiratory droplets, do the VOCs have potential for a fast triage type test? And actually, one of the things that was interesting and published quite early on in the COVID pandemic was people that even when they have no symptoms or very mild disease, when they did CT scans, so chest scans of those people, you still saw damage in the lungs. And actually, if you have damage in the lungs, that means that you're likely to be producing volatile biomarkers or markers of oxidative stress, inflammation as well. So there may be the opportunity to use breath testing in a very fast screening. It wouldn't be super specific, but again, as an initial first pass, that then you could send someone for follow-on PCR analysis is possible. We're not doing a lot on that, to be honest, because like the way we see it is that COVID's obviously horrendous. The PCR tests are good. They work well now. Once the world hopefully gets back to normal in not too distant future, the thing that won't have changed is 1 in 2 people will still get cancer, and the vast majority of them will still be diagnosed with late-stage disease. So we're trying to stay really, really focused on that problem as well.
A question from several people, in fact, is about one particular cancer, and that's pancreatic cancer. I wonder if that was something that the system would actually detect, because I know from personal experience that when it does get hold of somebody, it goes through them very, very quickly.
Yeah. No, it's an awful cancer, and it's very few cases are picked up early in it. I think one of the things that makes it quite tricky. So if you assume that there were some volatile biomarkers associated with the cancer, one of the things that makes it difficult is it's very deadly, but it's relatively rare. So compared to something like colorectal cancer or lung cancer, and that actually means that you need to have pretty spectacular performance. Otherwise, you'd have way too many false positives, for instance. So I don't think there's the same types of clear screening guidelines that are in place for that. So that's one of those ones where we're very keen to work closely with the academic partners. So the paper I showed earlier was a pancreatic cancer paper. And that's about trying to prove out is there some DOCs there or not, and if there is, then maybe that's something that gets folded into one of the core development activities. But lung is going to be the main priority for us for the near term.
Okay, and no time's getting on, and Phil will sort of come up with a time in a moment. But can this test be coupled with other tests like lung function, or will gases interfere with the test?
No, I think taking a multimodal approach or combining it makes a lot of sense, particularly in things like early cancer detection. Liquid biopsy, I think, has certain challenges, but it equally you could well imagine where you combine breath biopsy and liquid biopsy, or you have an initial breath biopsy test followed by a CT scan, for instance. So I think there's a lot of scope, to combine tests.
Okay. One question is, do Owlstone offer placements for students who would like to sort of work with your good selves?
We do. We have students every year, so absolutely. We myself and the other co-founders, we got a lot of support, from people within the kind of local Cambridge community. We think it's really important to try and support the engineers and scientists of tomorrow. We've sponsored PhD students in the past, and actually, a lot of the time they come and work for us afterwards. I think we've done like 3 or 4 PhD students. Then we have placements from students every year as well.
Okay. How long does a sensor last before it is polluted by previous samples?
The silicon device is actually pretty robust. So one of the things that we do is control how much it goes in at the inlet, because actually it's very, very sensitive. It's a common problem to poison sensors if you overload them. But one of the nice things is that unlike metal oxide or ENOS type sensors, where the compound comes in and it sticks to the surface and modifies a property like the resistance or something like that, that causes poisoning and contamination drift often in those types of sensors. In our device, it's a solid state filter. So the ions which are coming in, you're filtering them, you tune it for the ion of interest. But if you get a deposit, for instance, you just heat the sensor up basically. So you can raise the temperature of the sensor. So we tend to run the whole region at an elevated temperature, and that tends to stop contamination. Like I think the only, like for super sticky compounds, you need to be a bit more careful. But for anything that's pretty volatile, then you don't get any memory effect or contamination or hang up problems.
Okay. Time is coming on, but how does breath limonene testing compare with conventional blood liver function tests in terms of specificity and sensitivity?
That's a good question. Actually, the one of the, well, a couple of the guys in the company, one of the things that they say is actually what the blood tests are measuring isn't function. They're called liver function tests, but actually they're markers of damage. So with the approach that we're taking with limonene and evoke probes, it is a true measure of function there. So in the paper that we published, we've correlated everything with those blood-based markers. Actually, so that's all. It's up on our website in the publication. But I think it's a slightly different take in the sense that they are actually damage markers, not really function markers.
Okay. It looks like Phil's here, so the time is getting near to the end, and when you're on Q&A, time flies. So thank you very much, Billy, and I'll hand you back to Phil.
Thanks, David.
Yes, thank you, David. And Billy, there are still a few questions, quite a few questions left over. So appreciate your offer to answer those offline later.
Yeah, I'll get to those.
Billy, thank you. It's been a fascinating presentation on a groundbreaking technology and something that could help so many people potentially in the future. And thank you for sharing your personal journey and your obvious passion for the technology as well. It's good to see.
Yeah, no, thanks for having me. Really appreciate it.
It's been a pleasure. Okay, so that brings us to the end of tonight's event. So thank you all for your time and for joining us tonight. Please keep an eye out for our future events, which you can find by looking at the IET Cambridge website. And as I said earlier, you'll be able to find this recorded presentation on YouTube if you search for IET Cambridge. So on behalf of the IET Cambridge network, I'll thank you again for joining, and I'll bring this event to a close. I'll leave you with the link to the survey which the IET are doing on diversity and inclusion. Thank you, Billy. Thank you, everyone.
Thanks all. Bye.